Okay, so now you're familiar with the clinical samples that you will be working with over the course of these 3 crunches during the machine learning challenge. And now let's look at how the data is structured and how it will be provided to you. So for the spatial transcriptomics measurements, here we're showing tissue sections sitting on the Xenium slide. And here we're showing the spatial transcriptomics data itself, all 480 genes, where each dot is a transcript or one of those 480 genes. And on the right, we're showing the corresponding H&E image. And so you will be getting paired data like this for all 8 of these clinical samples. And now let's talk a little bit about how we chose to design these experiments. So at some point you might have wondered, well, Or is saying we've measured 480 genes here. How are those 480 genes chosen? Remember, there's 20,000 genes in the human genome, and we didn't just flip coins to choose these genes. Actually, what we did was we looked, first of all, for genes that are specifically expressed in cell types found in the colon. So a gene like EpCAM, which you remember marks epithelial cells, that's specifically expressed in epithelial cells in the colon, and is going to tell us about those types of cells. So that's a gene we chose. So we did that for many, many cell types and chose genes that mark those cell types. We also chose genes that have a known genetic association with ulcerative colitis. And what that means is that patients who have ulcerative colitis, they tend to have specific genetic variants that increase their risk for ulcerative colitis. So we think those specific genes are important in the pathogenesis, the disease mechanism of ulcerative colitis. Finally, we chose genes that are involved in cell-to-cell signaling. So this is what cell-to-cell signaling is really is just one cell sends a signal to another cell and that cell responds. And that's something that spatial transcriptomics gives us unique power to look at, because to look at a cell that's sending a signal and a cell that's receiving a signal, we really want to know where they are in space. So this cell signaling is very important to us, and we know that cell signaling drives inflammation. So that's another set of genes that we kind of pulled from, or ones involved in cell-cell signaling. And then the next question you have is, okay, what are the data types provided? So we're going to give you histology images prepared by us. We're also going to give you the Xenium data itself, the Xenium spatial transcriptomic data. That will be gene transcript locations and the assignment of those transcripts to cells. And then finally, we're going to give you the segmentation masks. And the segmentation mask, to remind you, is going to be a boundary that goes around every single cell, and it will tell us where the cells are and which transcripts are in those cells. And ultimately, for every tissue that we give you, every tissue section, there's hundreds of thousands of cells. Like typically, there's 300,000 or 400,000 cells that are being profiled in a single Xenium spatial transcriptomic data set. Okay, so how is the data going to be provided to you? So we've structured it using open source Python packages. So Xenium data is actually quite complicated. There's many data types that are needed to fully express the data. So we're going to be providing to you the data in an object that can be opened in the spatial data Python package. And spatial data is a really new framework that came out this year for the storage and process of multimodal spatial omics data. And this allows you to store images, points, labels, shapes, and tables. So images would be things like the H&E image. Points would be like the transcript location. Shapes will be the segmentation masks. And labels and tables are essentially metadata that we can use to annotate the features in the spatial transcriptomics data. And so spatial data is really it's really a package meant for reading in the data and organizing it in this really beautiful modular way. And if you want to do analysis on the data, there's another package that works seamlessly with spatial data called SquidPy, and we encourage you to check that out as well. So that's how the data will be structured and organized, and you'll get these 8 spatial data objects, one for each clinical sample. Now there's also the single-cell transcriptomics data, and there's 2 single-cell transcriptomics data sets we're going to be providing you with, one for Crunch 2 and one for Crunch 3. And remember, the single-cell transcriptomics data is going to give us expression measurements for all 20,000 genes, and we know that different cell types express different gene expression programs. So one data set we'll be giving you is one that we already introduced from Lecture 2. This is the one collected by Dr. Ron McXavier, where his group profiled ulcerative colitis tissue and did single-cell transcriptomics. And so there's information on immune cells and epithelial cells and stromal cells, which make up connective tissue in the colon. We will also provide you data sets collected from colon muscle and the colon nervous system. And we provide these as well because these represent cell types that are not found in this first single-cell transcriptomic data set. So with these data sets, you'll have representatives of all cell types that are found in ulcerative colitis colon. We'll also provide you with metadata for every single sample, and this metadata tells us whether the tissue came from a healthy individual or whether it came from a non-inflamed part of the colon or an inflamed part of the colon. And this is helpful because you are probably going to be most interested in those pieces that are from the inflamed colon, because that's going to tell us about the inflammation programs. But having the healthy there provides a point of comparison, so you can really look at what genes are differentiating inflamed from healthy colon. And then the last single-cell transcriptomics data set we're going to provide you comes from a study that was made looking at the colorectal cancer continuum. So this was a study carried out by William Greenleaf and Michael Snyder at Stanford University, and they collected samples from healthy colon, from adenomatous polyps or precancer, and from colorectal cancer. So polyps are abnormal growths of the colon epithelial cell lining into the center of the colon tube. And sometimes they look like mushrooms, or kind of just these projections inward into the colon. And so the polyps themselves also have dysplasia in them. So within the polyps, there's usually or often abnormal growths of epithelial cells. So this is a good data set where we can actually look at what dysplasia looks like at the single-cell transcriptomic level. So in their data, they collected samples from healthy colon, from colorectal cancer, and from these polyps, which often have dysplasia in them. And we have epithelial cells, immune cells, and these stromal cells, which again make up the connective tissue. And we have a lot of metadata provided here. First, we have what's the status of the tissue, whether there's a polyp, whether it's fully progressed to cancer, it's an adenocarcinoma, a type of cancer, or whether it's just healthy, so we call that normal or unaffected. In addition, we have the diagnosis of dysplasia from a pathologist, so we know which pieces of tissue have dysplasia in them, and that's going to be important for Crunch 3, where we're focused on dysplasia. And then finally, one thing you might want to focus on as you use this data is to focus on those polyps where there is diagnosed dysplasia by the pathologist. So that's going to give you single-cell data from dysplasia samples. So when you're working with single-cell data, there's a lot of packages out there, but we have structured the single-cell transcriptomic data such that you can load it again in another open-source Python package called ANData. This is a way to store the data, the gene expression count matrices, in matrices, and also have annotation for those matrices. And then there's another package called ScanPy, which you can use to carry out analysis of the different single-cell data sets. And so this is going to be very useful for working with the single-cell data. And what's nice is these packages all cooperate very well with the spatial data package that I introduced to you a few slides earlier, which you're going to use to work on with the spatial transcriptomic data.